Our next speaker is Dr. Matthew Ellenwood. He's a comparative medical genetics researcher at Iowa State University, where he works with a special focus on MPS I and MPS IIIB, using canine and mouse models to better understand the disease and to treat patients. He also chairs the Society's Scientific Advisory Board, and he's going to be speaking on gene therapy and gene editing both today and in the future. Welcome, Matthew.
I'd like to express my thanks to the Society for asking me to help with this year's family conference and the opportunity to present today. And thank you, Lauren, for that great primer. It sets up a lot of things for me in many respects. I'm mindful that I have a big task. We have families here who may be newly diagnosed. We have families here who may be professionals in research and medicine and extremely nuanced in their understanding of these things. So I'm trying to pitch the topic so that it'll be available to everybody. So topic is gene therapy for MPS disorders. Let me see here. So we'll have an overview of all of the current gene therapy trials that I'm able to find. There are so many now. When I started and first presented, I think it was in 2001, and I think Lauren actually introduced my talk at the MPS meeting that Liz Neufeld hosted then. There were no gene therapy trials. I was able to count 17 current, ongoing, or concluded gene therapy trials. This is a great time for us. I will concentrate on the background relevant to those trials that are active at this point. I will close with some potential therapies. There is a lot of buzz these days about gene editing. I will give that not very much attention right now, but I want to talk about some of the things we need to think about in terms of exploiting that approach. Over the development of, so the way I define gene therapy is pretty broad, and we've had to extend our definition as more approaches come up. A therapy that confers nucleic acid to a cell and or directly targets cell nucleic acid, and a therapy that augments, edits, or otherwise changes gene expression to achieve some therapeutic goal. In the background of gene therapy are a couple of historic events to the first decade in this century, and they both involved deaths or extreme illness in patients. One was a gene therapy for a severe combined immunodeficient disease conducted in France, where the gene therapy was directly implicated in cancers in the patients. The other involved something called an adeno-associated viral vector that, both due to enrollment concerns and deficiencies as well as preclinical work in large animal, led to the death of a patient. And that put the brakes on a lot of this work, but it also highlighted safety. So I think in some respects, while it's deeply regrettable these things occurred, we have a very much more cautious approach to the way we view things. Gene therapy in the MPS disorders, I think, has had a really exciting prominence, and there have been a couple of reasons historically. Many of our diseases are neuropathic, and the central nervous system disease has particular issues about treatment that make gene therapy attractive. And basically, I'm talking about the blood-brain barrier and the ability to deliver gene therapy directly to the brain or in some way target the brain makes it an attractive target. Most of our diseases, except for MPS IIIC, are caused by a soluble enzyme. So cross-correction is a feasibility for our diseases, where we can have a depot of production within a body and it can be secreted and taken up. We also may have a very low threshold for clearance of storage. And while I would agree with almost everything that Lauren said, I would say that our MPS diseases are not just storage diseases. But certainly the inciting cause is storage. But Lauren hit the nail on the head. There are so many new cascades out there that we are discovering that are common to many other diseases. But at least correcting that inciting cause, there may be a very low threshold that we need to reach. Vectors. You may have heard about vectors. Now, if you're thinking geometry or engineering, I'm not talking about lines, right? We're talking about things that will take a gene therapy into a body, into a cell type. Most of these are genetically engineered viruses, and they're used to create some therapeutic effect. One of the most popular is something called AAV. It is a viral gene therapy vector. Did I miss a slide here? Yes, I'm sorry. So vectors just deliver nucleic acid to a cell. There are non-viral vectors. These are not stuff I'm going to talk about today. Among the most common and safe vectors out there, and a vector that is in clinical use now with other diseases, is AAV. That stands for adeno-associated viral vector. These are very small viruses. They're very sturdy. They're easy to produce. They're related to parvovirus, which is in some respects a similar virus to the one that may potentially infect your dog or for which you get your dog vaccinated. AAV, however, are defective viruses. They require in the wild a second virus to reproduce themselves. That makes them a safe selection. Furthermore, when we genetically engineer them, we take out all their regular genes and replace them with our gene therapy payload. And that vector is fully replication incompetent. It can't divide in cells. And that, again, makes them very safe. These are non-integrating vectors. They do not go into the genome of the patient. As I said, production and purification is relatively easy. They have these coat proteins, and you can see, whoops, I'm not sure I can get that. That top illustration is an image of a viral model, and those little nubbins, those are protein coats. They will bind to certain cells, and that gives the vector some cell specificity, or what we call tropism. We can genetically manipulate that, and there are multiple what we call serotypes, and they're all changing out that coat protein that's used to bind to certain viruses. So if you've heard of AAV9 or AAV8 or AAV2, it's all referring to the different capsid protein or coat protein, and it will give those vectors a different cell target. It is not absolute. You will get some spillover to other tissue types. It does give us tremendous flexibility with these vectors, which makes them very attractive. The vectors have a relatively small genome. Fortunately, so do most of our MPS disorders in terms of the size of the cDNA or the processed gene that we need to insert. So all of them except for MPS IIIC will fit nicely into that kind of a vector. Another vector that's being used currently or being evaluated is what we call a lentiviral vector. Now that may make you sit up and pay attention. There are lentiviruses that are extremely concerning medically. HIV is a lentiviral vector. Lenti just means slow, and they are a slowly developing retroviral disease. The beauty of these things is we can genetically engineer them. That means take out all the pathogenic viral proteins, insert our payload, and we can get a payload of all of our MPS genes in here, and we can do some genetic manipulations to make these guys much safer. And then they can be used to transduce cells. They integrate permanently. In other words, they put DNA right into the genome of the patient cells. And they can, there were, as I said, concerns in the past about safety. These involved using the proper gene promoter to drive expression. Those have been corrected in application these days. And there were also some viral promoters, what we call long terminal repeats, and they have been eliminated. These are so-called self-inactivating vectors. So there's a much higher threshold of, or a safety profile for these now. Their use in human medicine is to transduce or to infect cells in vivo or in vitro, stem cells that we've acquired from a patient. We transduce them in the Petri dish, and then we'll inject them back into patients. That tends to be their usage. There are also hybrid approaches involving mechanisms to edit genes that are also being targeted with gene therapy vectors. So they may change the native sequence in a targeted way, and targeted is the important thing when we talk about editing. They may be trying to insert the gene into what we call safe harbors, which are areas of the genome we know we can get genes into, or they may be trying to target a highly expressed locus in an important gene. So this is the approach Sangamo Therapeutics uses. They put the cDNA for MPS I and MPS II into what we call an albumin locus. Albumin is the most highly expressed liver in the protein. We can sit that gene right in front of the promoter, which says produce lots of iduronate sulfatase or produce lots of iduronidase, and we can get targeting that way. And again, a hybrid approach also involves targeting cells outside the body, hematopoietic cells. We're not treating the patient directly with gene therapy, we're treating their cells and then giving them back to the patient. So I'm going to go through the 17 or so trials. My disclosure statement is all of the errors up here are mine and may not reflect the numbers that are really out there. Juggling all these things, I hope I didn't get anything wrong. So Abeona Therapeutics, they have a product, ABO-101, to treat MPS IIIB. This is an intravenous therapy targeting the cells that line the brain circulation. So it's a brain-targeted therapy, but it's intravenous administration. They are using an AAV9 vector, so that's adeno-associated virus serotype 9. They are recruiting 9 patients. This trial is underway, started in 2017, estimated to be finished 2020. They were using 2 doses of the vector, so patients got 2 different doses to evaluate a dose response. I also want to highlight this number here, which is a clinical trial number. And I'll go through the kind of information that you can find for these sorts of things. You can go to the clinicaltrials.gov site and just type in mucopolysaccharidosis and gene therapy, and you'll get a list of all the really granular detail that you might like to have. And it's really quite thorough. So details for this, there are 9 patients. As I said, it's a dose escalation. Most of the trials, except I know one, has progressed to phase 3, are combined phase 1 and 2. Their primary endpoints are looking at safety, but they will evaluate some elements of efficacy. They're dosing patients with 2 different doses. So there is the dose 2 times 10 to the 13th. And that's viral genomes per kilogram. That's a really, really big number when you start to think about it. They're transferring a recombinant AAV9, and it's transmitting the cDNA for the missing gene, NAGLU, and those letters in front of it refer to the promoter, or the DNA sequence that tells the gene when to run. This is a one-time infusion intravenously via peripheral limb, start dates, completion dates. It's a multi-center trial, and the criteria for entry was MPS IIIB greater than 6 months with neurological dysfunction. You'll be able to find that level of detail through all these clinical trials. Full disclosure, I just chose the first company by alphabet and the first trial. I'm not trying to plug them. You'll get all this detail for the others. So similarly, Abeona has a trial for MPS IIIA. Again, same level of phase, same vector roughly, except of course it has a different payload. It has the gene that's missing in MPS IIIA, which is a sulfatase. And there is a criteria for entry. Patients needed to be ambulatory and a developmental quotient less than 60, and single dose therapy for this. There's also a trial based in Naples. This is a multi-institutional trial. This is one of the few trials for a primarily bone disease. This is MPS 6. It is liver-directed, so they're looking to transduce the liver and express enzyme from that will be distributed through the body by circulation. And those are the dates and the levels. And again, when you see vectors that say AAV2/8, that means it's serotype 8, but the DNA that's part of the vector has the ancestral components to from AAV2. And in fact, almost all the vectors, as far as I know, have that 2 component. Some labs will list 2/8, some will just list 8. Other trials we have a single institution trial in Milano. This is an interesting trial, and this is a therapy that the group at Milan have pioneered for severe combined immunodeficiencies, and it's been very successful in that area. Those diseases are different, though. If you can get a couple of gene therapy-corrected cells for an immunodeficiency, there's a selective advantage because they're functioning immune cells, and they will do really well. It's a bit different in MPSs where we are transducing bone marrow and maybe not completely, but it's still a very exciting and proven approach. So they're taking autologous hematopoietic stem cells. So they'll take hematopoietic stem cells out of a patient, they will transduce them with a lentiviral vector, which I described earlier. And then they'll infuse those back into the patient who has had some kind of what we call conditioning, preparing their body to receive bone marrow transplantation or hematopoietic stem cell transplantation. In the bad old days, that would be full body irradiation, but now we know how to do it in a much more nuanced way involving chemotherapy to limit the downside effects on the patients. So this is being pursued for MPS I and very exciting approach. Again, just in Milan. We have Lysogene, which is pursuing a intraparenchymal AAV therapy for Sanfilippo A. So they're actually injecting the vector into the brain tissue as an approach to getting around the blood-brain barrier. And they have had 2 trials, both with MPS IIIA. The earlier trial listed there, is that my time? Oh, we're good, okay. That trial's concluded. There was a wide variety or variability in the age of which patients were recruited. There were no adverse events, and there was enough positive results to continue with a second trial with a slightly changed vector. And I know Lysogene has representatives here, and I'm sure they'll be excited to discuss a lot of the details about this trial. It's ongoing. I think it's not fully enrolled yet, from what I was told. And they're using, again, AAV10. And this is a human-derived AAV serotype. So you'll see RH in front of the AAV. And that's an AAV serotype that's supposed to be particularly good at transducing or delivering genes to the brain of human patients. I'm glad I looked things up to double check. Manchester just announced and posted a trial on the 15th of this month that is multi-institutional for autologous hematopoietic stem cell transduction using a lentiviral vector system. And this is for 3A. So the idea is that bone marrow cells in the patient will differentiate and become, in some cases, the type of cell that may migrate to the brain and set up as an immune surveillance cell in the brain. And by overexpressing the enzyme in these cells, we can get a depot of enzyme migrating to the brain to treat these patients. My work is in MPS III. I'm really excited to see where how this study progresses. There is a historical trial listed on the site. You'll see that happened at University of Minnesota back in the 90s, and that was a very early study where they were just looking at transducing peripheral blood lymphocytes that you'd take out of somebody's arm with a retroviral vector, but it did not go any further than that. Regenex Bio is got 2 trials ongoing that are very exciting. These are for the treatment of MPS I and MPS II. They are using an AAV9, which seems to do a very good job of transducing central nervous system tissue, and they're looking at an intrathecal dose administration via the cerebrospinal fluid. And this is a biotech company that has long been associated with researchers at the University of Pennsylvania. There is a gene therapy trial at St. Jude's that is only involving long-term marking studies, and the way I read it, it was just for MPS patients treated at St. Jude's, and I don't think they have any gene therapy trials for MPS I there, but it will show up. Sangamo Therapeutics. I think I'm on the 4th or 5th slide of gene therapy trials. This would have been an unheard of event even 5 years ago. Sangamo Therapeutics is a really interesting approach to treating the MPSs. They're looking to treat patients, primarily adult, with MPS I and MPS II, and they're trying to stick the normal gene into the gene of a highly expressed liver protein, and using that endogenous protein promoter or gene promoter to drive expression. They're basically trying to hijack a certain number of cells in the liver and turn it into a factory to produce enzyme. So they've got very specific, what we call nucleases. They cut the genome like almost a scissor in very precise spots, and we get the genome inserted into those positions, the gene therapy vector genome. So they've got products for MPS 1 and MPS 2. And the attractive thing about this is it will be an endogenously produced enzyme with a pretty strong level of therapeutic safety and efficacy based on animal studies. Finally, wrapping things up, there's been a study done in Paris. This went on in parallel to the early study at Lysogene, targeting, again, intraparenchymal or right into the brain tissue injection of AAV vector to treat MPS IIIB. This study is concluded. There were no adverse events, and they are not yet recruiting more or expanding this study at this point, as far as I'm aware. So broadly speaking, we have results that are encouraging for further evaluation, but we also have a long way to go. In none of these have we seen a home run, but we've also seen no serious events of the sort that marked some of the early gene therapy history in the research community. Continued concerns will be age at enrollment. Are we able to treat individuals at that therapeutic window, as it were, before the water, to use Lauren's analogy, has started to spill over the rim of the bathtub? We also need to refine our dose delivery. Scaling up from mice to humans is very difficult. The large animal models have proved very important for that. They've been very useful, for example, launching gene therapy in the gangliosidosis in the cats, etc. We also have continued issues unrelated necessarily to gene therapy. You'll hear more about newborn screening, I think, with Barbara speaking. The big issue there is getting a conclusive diagnosis and a conclusive prognosis to know if your patient needs to have a gene therapy. Reaching the brain is a real concern. And finding improved predictive endpoints for slowly progressive disease. If you go through and read these gene therapy prospectuses on clinicaltrials.gov, the primary endpoints are fairly limited, and a lot of them don't necessarily impact things that we may find necessarily important, but they're things that we can measure in a discrete period of time that could lead to approval. Improving our understanding of imaging, improving our understanding of psychiometry testing, and Julie will talk about that a little bit later, is really critical so that we have an improved understanding of disease progression and we can predict this patient would have progressed to this state if they had not had gene therapy intervention. So future prospects I'm quite excited about involve combining things even further. So thinking about a liver-targeted therapy, maybe of the likes of Sangamo, where we actually have a fusion protein of an enzyme that has a tag on it that may somehow lead to it being taken up by the brain. So you'd have a liver-targeted gene editing that leads to a central nervous system improvement. What we haven't talked much about is bone targeting. The issues of bone disease are complex and clearly involve more than just enzyme activity, but we also have concerns about if we have a window of opportunity, do we have the ability to get enzyme into what's a fairly avascular space? Can we do things to improve vector targeting to the joints of these patients? You will have heard about gene editing technology, CRISPR and the like. There's now more than just CRISPR. These are very exciting, but I think our excitement needs to be a bit restrained. We have to think about the target organ we are going to be focusing on. How much of that are we going to be able to correct? What is the therapeutic threshold? The ability for these gene editing technologies to transduce or to transform cells is well below the 5% sort of efficacy rate that we would expect to start to see a therapeutic effect. And that's just for the whole body and not necessarily for one organ. So we're really concerned about developing improved editing efficiencies. There's also concern about off-target editing. You may be targeting editing in the liver in one gene, but you may get editing in some other gene that has other problems associated with it. The studies that I'm aware of make off-target editing less of a concern than just random mutations. All of us have inherited from our parents roughly 60 mutations that weren't there in our folks, and the off-target mutations seem to be about at that level, so that may be less of a concern. I didn't know we had a panel, so I will skip through questions and wait for the panel time, and I hope I have not gone over.